Scientists have printed functional brain tissue. The world's first functional brain tissue produced using a 3D printer was created at the University of Wisconsin-Madison. This tissue can grow and develop in the same way as natural brain tissue. This achievement has important implications for scientists studying the brain and developing treatments for a wide range of neurological and neurodevelopmental disorders, such as Alzheimer's and Parkinson's diseases. Three-dimensional printing technology is used for many things. It is used to create houses, prostheses, implants and even organs such as the heart. Bioprinting technology has developed significantly in recent years. As new biomaterials for producing bioinks are developed, 3D printers have gained completely new possibilities. Thanks to this, researchers were able to produce tissues and even models of human organs. Now it has been possible to print brain tissue that can grow and form connections in the same way as real human brain tissue. The results and description of the research were published in the journal, Cell Stem Cell. The University of Wisconsin-Madison team's achievement gives neuroscientists a new tool for studying communication between brain cells and other parts of the human brain, potentially leading to better treatments for neurodegenerative diseases. This may change the way we look at stem cell biology, neuroscience and the pathogenesis of many neurological and psychiatric disorders, says Su-Chun Zhang, one of the authors of the publication. According to Zhang, it was the printing methods used that limited the success of previous attempts to print brain tissue. In the new work, instead of the traditional approach to 3D printing, which involves stacking layers vertically, they decided to arrange them horizontally. They placed brain cells, neurons grown from induced pluripotent stem cells, in a bioink gel. However, the bioink used in these studies had a slightly different consistency and was softer than the inks used in previous trials. Tissue printed from such bioink retains its structure and is delicate enough to allow neurons to fuse with each other and start communicating. Our tissue remains relatively thin, which makes it easier for neurons to obtain enough oxygen and nutrients from the growth medium, says Yuanwei Yan from Zhang's lab. Printed cells can do more than just communicate with each other. They create connections within each printed layer, as well as between them. This creates a network comparable to human brains. Neurons communicate, send signals, interact with each other via neurotransmitters, and even form appropriate networks with support cells added to the printed tissue. We printed the cerebral cortex and striatum and what we found was quite striking. Even when we printed different cells belonging to different parts of the brain, they were still able to communicate with each other in a very specific way, says Zhang. This is thanks to the precision provided by the new printing technique, which offers control over cell type and arrangement, unheard of in cerebral organoids, miniature organs used to study the brain. Organoids grow with less organization and control. We have developed a 3D bioprinting platform for assembling tissues with defined human nerve cell types to the desired dimensions using a commercial bioprinter. The printed neural progenitors differentiate into neurons and form functional neural circuits within and between tissue layers within weeks. Printed astrocyte progenitors develop into mature astrocytes with complex processes and create functional neuron astrocyte networks, the study authors wrote in the publication. They added that such tissue will be useful for brain research, better understanding of the operation of neural networks, and modeling of pathological processes. It will also serve as a drug testing platform. We are able to produce virtually any type of neurons at any time. We can assemble them any way we want. Because we can print the tissue as designed, we can have a defined system for checking how the human brain network works. We can look very closely at how nerve cells talk to each other under certain conditions because we can print exactly what we want, emphasizes Zhang. Printed brain tissue can be used to study signaling between cells in Down syndrome, interactions between healthy tissue and adjacent tissue affected by Alzheimer's disease, test new drug candidates, and even observe brain growth. Most bioprinting strategies focus on mimicking fully mature tissues. In the future, bioprinting methods may take into account earlier developmental stages. The new technique does not require special equipment or culture methods to maintain healthy tissue, so it should be available to many laboratories. Additionally, such tissue can be examined using microscopes and other equipment typically found in most laboratories. The pig's brain was kept alive outside the body for 5 hours. Using a special device, scientists managed to keep a pig's brain alive outside its body for 5 hours. Such a device could allow for new ways of studying the brain without affecting other body functions. A device that keeps the brain alive and allows it to function separately from the body was created at the University of Texas Southwestern Medical Center, UTSW. They were tested on pig brain. Using it, scientists managed to keep a pig brain alive for 5 hours. This could lead to new ways of studying the human brain. Access to a brain isolated from the rest of the body is desirable for metabolic, pharmacological, and other studies aimed at characterizing neuronal properties in isolation from influences from the rest of the body. The results and description of the research were published in the journal, Scientific Reports. The brain is responsible for a variety of processes that regulate, among others, heart rate, breathing and sleep-wake cycles. But its function is influenced by factors from the rest of the body, such as blood sugar levels, blood pressure and blood oxygenation. Scientists from UTSW are not the first. In 2018, scientists from Yale University also managed to keep a pig's brain alive outside the animal's body for much longer. Other research teams have also attempted to keep the brain of a monkey or dog outside the body. The newly developed system allows you to isolate the brain from the rest of the body using an intelligent artificial pump that takes care of its blood supply. To mimic real circulation, the device can subtly modify blood composition and adjust blood flow to a number of variables, including pressure, volume, temperature, oxygenation and nutrients. This is possible thanks to a special algorithm that monitors blood pressure, flow, pulsation, and other factors. In the study, the system was tested on the brains of domestic pigs. First, the blood supply to the pig brain was surgically separated from the systemic circulation and full extracorporeal circulation control was provided. This control was based on a developed algorithm that maintained blood pressure, flow and pulsation at levels close to those measured in this particular pig before the experiments began for several hours. Once connected to the device, brain activity was monitored using electrocorticography. This is a type of electroencephalography in which electrodes are placed directly on the cerebral cortex. Interestingly, the pig's brain activity remained unchanged or minimally affected outside the body. Isolating the brain will allow scientists to manipulate the input signals reaching it. This will allow us to examine how they change brain function without affecting the body. Juan Pascual, who led the study, said he and his colleagues used the system to better understand the effects of hypoglycemia, low blood sugar, in the absence of other factors. Although scientists can induce hypoglycemia by restricting food intake in laboratory animals or giving them insulin, the body can partially compensate for either scenario by changing its metabolism. The device removes these restrictions. No body, no problem. Scientists can change the glucose content directly in the blood pumped to the brain. This novel method enables analyses that focus on the brain independently of the body, allowing us to study the brain in a way that has never been done before, Pascual said.